Okay, so today we're going to start talking about cell division, and our focus question is how do we grow and repair wounds? So, for cell division, this is usually used for growth, repair, or replacement of cells. And the way that we do this is through a process called mitosis, where one diploid cell becomes two diploid cells. And when we're talking about diploid, we're talking about the number of chromosomes. So, this is in any somatic cell or any cell that is not a gamete. So there's gametes and there's somatic cells. And somatic cells are any cells in your body that are not an egg or a sperm cell. So your liver cells, your heart cells, anything like that is considered a somatic cell. And in mitosis, the daughter cells have the same number of chromosomes as the parent cells. So there's 4 phases we're going to talk about. And the first one's going to be prophase, and then metaphase, anaphase, and telophase. And within telophase, we are also going to talk about a process called cytokinesis. So in prophase, the DNA replicates and condenses, and the chromosomes start to appear, and the nuclear envelope that goes around the nucleus starts to disappear. You'll also notice that the spindle apparatus is starting to form. In metaphase, the chromosomes line up on what we call the metaphase plate, and they create this line across the middle, and the spindle apparatus is then connecting to the chromosomes as they're lining up. In anaphase, the chromosomes are pulled apart by the spindle apparatus. So anaphase apart, and you'll notice that they're starting to pull down the spindle apparatus towards the poles or the ends. And in telophase, the chromosomes start to unravel and the nuclear envelope reappears and the cell starts to split into 2 cells. So in an animal cell, you'll see this area right here called the cleavage furrow, but in a plant cell, you might just see the formation of a cell wall, which is one of the differences between the two. Now, real quick to look at this word cytokinesis. So cyto is talking about the cytoplasm, which you all know is this fluid that kind of surrounds the organelles. And when it talks about kinesis, it's talking about splitting up. So this would be splitting up the cytoplasm or the actual division of the 2 cells. You guys are going to watch an animation of mitosis, and this will give you a little bit of a better idea of what's going on. So I'm going to play it for you on here.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between 2 daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into 2 daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, so you guys can get started on your extension work and finish your worksheet for the day. All right.